Center for the Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on testing for lipids. In our last lesson, we learned about testing for proteins. We studied the Biuret test, which indicates peptide links using a Biuret reagent. We also briefly studied the structure and functions of proteins and the formation of amino acids. Today, we are going to learn about tests that determine the presence of lipids in a substance. We will begin the lesson by discussing the structure and functions of lipids, and then learn about the grease spot test and emulsion test. Let us begin. Lipids are a diverse range of organic molecules that include fats, oils, waxes, phospholipids, and steroids. They are insoluble in water, but soluble in organic solvents. Lipids are storage molecules, which means they do not function or interfere with the other reactions that occur in the cytoplasm. Rather, they store up energy in the body, which can be used when it is required. Lipids contain more energy per gram than carbohydrates or proteins. Fats and oils are a very important group of chemicals necessary for normal body functioning. Lipid-rich foods include any food that contains a large amount of fat or oil. Butter, sesame oil, olive oil, coconuts, and oily fish are examples of food sources that provide lipids. Fats and oils are chemically similar, yet at room temperature, fats are solids and oils are liquids. The lipids occurring in animals are more likely to be solid at room temperature than plant lipids. Students, before we move on to discuss the structure of lipids, please list as many characteristics of lipids as you can.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, everyone. How did you do? You may have said that lipids are insoluble in water, but soluble in other organic solvents. Lipids are storage molecules. Lipids contain a large amount of energy that the body requires. Lipids are found in plant or animal food sources. Lipids can be both solids and liquids at room temperature. Lipids can occur in the form of waxes, oils, or fats. If you listed any of these characteristics, you are correct. Great work. Like carbohydrates, lipids are made from carbon, hydrogen, and oxygen. However, there is much less oxygen in lipids than in carbohydrates. Lipids are made up of a combination of two types of organic chemicals, glycerol and fatty acids. Glycerol is always the same chemical. However, fatty acids can vary in two ways: by the length of the carbon chain and whether the fatty acid is saturated or unsaturated. In saturated fatty acids, the carbons are joined by a single covalent bond, and unsaturated fatty acids include double bonds. Single covalent bond fatty acids are not able to pack their molecules close together. Therefore, these types of fatty acids form lipids that are liquid at room temperature. When a molecule of glycerol combines with 3 fatty acids, a lipid is formed. Like carbohydrates and protein, the molecules link together in a condensation reaction, and a molecule of water is lost. We will learn about 2 tests that indicate the presence of lipids in a substance: the grease spot test and the emulsion test. The grease spot test is the simplest way to test the presence of lipids in a substance. Using a brown paper bag or a piece of paper, the substance that is being tested is put onto the paper with a cotton swab or an eyedropper. The substance is left to dry for a few minutes. If the substance contains lipids, the area where it was placed on the paper will be left permanently translucent. The translucent spot can best be seen when the paper is held against a light source, like a window. Students, let us do an activity. Pretend that you have 2 pieces of brown paper. On one piece of paper, you add a drop of olive oil. On the other piece of paper, you add a drop of water. Once you let both pieces of paper dry, what do you think they will look like? Write down your hypothesis, and I will see you in a few minutes.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Nice to see you again, students. If you said that the piece of paper with the olive oil would leave a translucent mark, and the piece of paper with the water would not leave a mark, then you answered correctly. Great job. The grease spot test is a good method to determine if lipids are present. However, this test cannot determine the amount of lipids in a substance. Typically, a chemical reaction is necessary to find out a comprehensive result in substances. The next test we will look at is the emulsion test. This test requires dry test tubes, ethanol, water, and a substance to test. In the test tube, equal parts of the ethanol and the substances are combined. Then, the test tube is shaken in order to dissolve the liquid into the ethanol. The contents of that test tube are poured into another test tube that contains water. If lipids are present in the solution, a white, cloudy layer or emulsion forms. Students, in the emulsion test, why do the lipids form a layer on top of the water?
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, everyone. If you said that the lipids form a layer on top of the water because lipids are insoluble in water and therefore cannot dissolve, you are correct. Good work. This brings us to the end of our lesson. Today, we learned about the grease spot test and the emulsion test to determine the presence of lipids in a substance. We also briefly discussed the structure and functions of lipids. Until next lesson, thank you, teacher. Thank you, students.